In this video, we are really going to shift gears, and now we're going to look at what are the cells, what are bacteria, or what are other organisms, how do we know how they're related to each other, how do we classify them, that kind of thing. There are two separate but related systems of information. Let's see, click. There. We have taxonomy and phylogeny, and they are, again, two separate things, but they kind of overlap, as you will see. So taxonomy is classification of organisms into groups. This is where we say this is this species, or this is that family, or that genus, or something like that. That's taxonomy. Phylogeny is the study of the evolutionary relationships between organisms. So phylogeny is where we say these two things are closely related or they are distant cousins. So taxonomy, we might make a statement like E. coli, Escherichia coli. E. coli cells are members of the phylum Proteobacterium. And phylogeny, we might say about E. coli. E. coli are more closely related to bacteria inside the Proteobacteria, so their phylum, than they are to bacteria from other phyla, right? And then we could also say at a totally different taxonomic level, we could say UPEC strains. These are strains of E. coli cells. These cause urinary tract infections. There are lots of strains of E. coli. The UPEC strains are all members of the E. coli species. That's a thing we could say. And then that's taxonomy. Phylogeny, we would say UPEC, E. coli, and EHEC, that's a different strain. UPEC, E. coli, and EHEC, E. coli are more closely related to each other, so members of the same species, than they are to strains of Yersinia pestis, which is an unrelated species that causes the bubonic plague. So again, taxonomy is putting the things into groups. Phylogeny is the evolutionary underpinnings of the groups. And the reason these are separate is that historically in microbiology, we didn't know the phylogeny. We had to do taxonomy without phylogeny. Now that we can do phylogeny, we have to change some of our taxonomy. Let's get into it. So in microbiology, we've always had to use taxonomy. We've always had to do things like identify the bacteria from a patient's wound to see if it's bad or see if it's one we recognize. And identifying it, putting it into a taxonomic group has always been important. So that's what clinicians would look at. Biologists want taxonomy to be based on phylogeny. We want the groups to be based on evolutionary relationships. But we weren't able to understand the evolutionary relationships between microorganisms until DNA sequencing in the 1980s, really. Right. So before DNA sequencing, we used everything we could to try to figure out phylogeny. We looked at the size and shape of cells, their behavior, their biochemistry, things like that. Unfortunately, those things don't track very much with evolution. Also, there are certain things that go on in microorganisms like horizontal gene transfer, which we will talk about later, that make phylogeny potentially unknowable. It is possible that we will never really be able to confidently say what the evolutionary history is of different bacterial strains. This is what phylogeny was built on for animals. So for mammals that all have the same bones, we can say, well, let's see, these animals these are more closely related to each other than this is to this one because, I don't know, the these two both have essentially, I'm just guessing, these look like carpals, like modified carpals and metacarpals as parts of the leg, whereas in a human, the carpals and metacarpals are just part of the hand. That's the kind of comparison we could do with things like bones, but microorganisms don't have bones. There's nothing we can examine with a microscope that tells us this kind of thing about microorganisms. In the 1980s, we started sequencing ribosomal RNA genes. So think back to what a ribosome is and what it's made of. It's made of protein and it's made of ribosomal RNA. And that ribosomal RNA is encoded by ribosomal RNA genes in the genome. So that's DNA with a sequence that is passed down from mother cell to daughter cell. And all cells that I'm aware of on Earth have ribosomal RNA. So we can compare the ribosomal RNA genes of all cells on Earth and construct phylogenies. What that means is we can try to figure out how they are related to each other. So we visualize phylogeny by building a phylogenetic tree or dendrogram. This is an example of the universal phylogeny. We'll come back to this many times. This is a good time for me to point out that if I show you a figure like this, I don't expect you to memorize everything in the figure. If I talk about this one and I talk about that one and I talk about that one, those you should know, but I don't expect you to be able to draw this from memory, or at least not with this level of detail. Not yet. So, we construct that type of phylogenetic tree using DNA sequences, and here's how that works. We get the DNA sequences of 4 different organisms. So this is an example. Let's say we have 4 different organisms, and we have their DNA sequences of their ribosomal RNA genes. In reality, so this is what 6 nucleotide bases, and we line them up. All of them start with A and then have a C, so adenine and cytosine. Some of them have another adenine, others have a thymine, whatever. Realistically, the 16S ribosomal RNA gene is 1,500 bases long. This is showing an example of a gene that is 6 bases long, but this can still show you how it works. What we do is we calculate the number of differences between pairs of sequences. So between 1 and 2, how many differences do we find? Well, no. So the same, different, same, different. So 2 differences between 1 and 2. So we make this table where we say between 1 and 2, there are 2 differences. Between 1 and 1, there are no differences. It's the same thing. And then between 2 and 3, so between 2 and 3, same, different. 4 differences. So between 2 and 3, there are 4 differences. So we make this table, and then we make a tree or dendrogram that has lengths that are these distances, or we do the best we can to make a tree where these lengths are these distances. It's complicated when you're using real genes that are 1,500 bases long. It's not straightforward like this with nice round numbers, and you have to build trees that are hard to build. That's what computers are for. So, how do we read these trees? Let me skip ahead to the next slide. How do we read them? How closely related are 1 and 3? Let's say that you only had this tree and you don't have the sequences. How do you read this? Well, 1 and 3, what we do is we find a path from 1 to 3. So we go like this, 1 to 3. And we count all the distance in this axis, this direction, horizontal distance. So 1, and then we ignore this. That's just a connector. And then another 1, and then we ignore this. It's just a connector. And then another 1, and then another 1. So that's 4 differences. So we'd say they are however closely related two things are that have 4 differences. Whereas 1 and 2 have 1, 2 differences. So they are more closely related than 1 is to 3. Take time to understand that before you go on. So go ahead, pause this, go back if you need to. Make sure you follow what I just said. Okay, so one point that comes up when we look at phylogenetic trees is these linkers. These don't have any information in them. So the fact that 1 is on top and 2 is on the bottom here is meaningless. What has meaning is that 1 is connected to 2. And this distance has meaning and this distance has meaning. But you could put 2 up here and put 1 down here, like I've done here, and it doesn't change the information in the tree. So it's the same tree, we've just drawn it a little bit differently. Same phylogenetic information drawn differently. So take some time to look at the differences between these two trees. I promise you they are the same information. They're both valid ways to show the same information. And this is what we do to understand phylogeny. So this is the universal phylogenetic tree. Notice we have eukaryotes over here that have animals. That's us. And fungi, that's molds and yeasts and mushrooms. All plants are here. And then everything else in this group, these are all the organisms that have nuclei and organelles. There are a whole bunch that are single-celled and super weird and distantly related to animals. So look at how close together we are to the fungi and the plants. All animals, fungi, and plants are more closely related to each other than we are to anything else. But yet we have all these other relatives that are eukaryotic. They have nuclei and organelles. And we say they are deep branching. They have these really long branch lengths before they get to this main branch that connects to us. Another way of looking at it is like fungi have this short branch. Fungi could be closely related to both plants and animals because there's not much distance from there to that common joining area. Diplomonads, like Giardia, have a really long branch. They're not closely related to anything else. They can't be because you have to measure this whole length before you measure whatever else. Okay, another thing we can see. So this is the domain of eukarya. There are 2 other domains of, these are both prokaryotes. So these are cells without, no nuclei. We have the bacteria. This is the most diverse group of organisms. Lots of long branches. And the archaea. They look just like bacteria, but they are weird, and we don't know them quite as well. In fact, we didn't realize that there was a difference between these and bacteria until 1977, when we started doing this ribosomal RNA sequencing, because any other way you look at it, these look the same as bacteria. We'll come back to this many times. So cool. So I've showed you what we do with DNA sequencing to make phylogenetic trees, and I've shown you this giant universal ribosomal RNA tree, and we'll come back to it many times. And later in the class, we will see what we use for taxonomy and what used to be done in research labs and what is still done in clinical labs, where taxonomy is more important than phylogeny. And really, phylogeny doesn't tell us very much about how cells behave. So, sorry, I'm skipping ahead to slide 76. Phylogeny doesn't tell us the behaviors of these organisms. So this is a tree of the, I think, let me see, yeah, all the proteobacteria. So this is all members of one phylum of bacteria. Escherichia is in here somewhere. Yeah, gamma. Escherichia, Vibrio cholerae causes cholera. Whatever. They're color coded by things like what type of metabolism they have, and notice how different the colors are. And the colors aren't like matched with two closely related organisms. So within this group, gamma proteobacteria that are all one group, more closely related to each other than they are to any other, there are tons of different types of metabolism. And so even if you know that some unknown bacterium is in this group, you don't know how it's going to behave. So phylogeny, we thought, would help us understand everything about bacteria once we could do DNA sequencing. But it turns out just knowing its evolutionary relationship to others doesn't tell you everything about it. Another interesting thing that happened when we started sequencing DNA is we realized that some of our taxonomy was wrong. So some of our classifications were wrong. So one famous example is two genera, genus Escherichia and genus Shigella. Shigella species cause dysentery, really bad diarrhea, sometimes with blood. So clinicians would identify bacteria as Shigella and know, oh, those cause these certain diseases, whereas Escherichia, which are closely related to them, many don't cause diseases. And then once we started doing DNA sequencing, what we realized is that they're all the same. They should all be called the same genus. They're all mixed together. So these are a bunch of Escherichia species in this group. And if you go out to here, they are closely joined to like this group of Shigella. And within this same group that has these Shigella, it has another group of Shigella, and on this side, Escherichia. If these really were different phylogenetic groups, these would all be Escherichia, these would all be Shigella. But there's no way to really separate them phylogenetically. They are one big group. It's just that a lot of the strains that do certain things have characteristics that we can identify. What I want to leave you with in this video is this simplified universal phylogeny, where we have a lot of branches that aren't even labeled, and that's fine. This is just another way of drawing that same ribosomal RNA universal phylogenetic tree, but it shows us certain things that we're going to want to focus on, and it doesn't show us a lot of extra stuff. So we're going to come back to this. But again, we have three different domains: eukaryotes, archaea, and bacteria, and major phyla within them, including the kingdoms that we have traditionally seen for things like animals, fungi, plants. How these are all related to each other is kind of a mystery. We don't really know exactly what the universal ancestor was. Biologists agree there probably was one universal ancestor of all cellular life on Earth, but what it looked like would it have looked like one of these? Would it have looked like one of these? We don't really know. And that is an active area of research. Indeed, people argue about the relationship between eukaryotes and archaea. Some people think this branch should actually go here, and eukaryotes and archaea are actually like together. But that's neither here nor there. And I'm going to end this video, and we will look at taxonomy next.